Good afternoon. Today I'm going to talk about the #1 cause of death in the United States and worldwide, and that's cardiovascular disease. It affects both men and women, as well as all races and ethnicities equally, and is a significant burden on society. So the most common type of cardiovascular disease is a heart attack. And this happens when a coronary artery, which supplies blood and oxygen to your downstream tissue, is blocked. There's cell death that's permanent that occurs within minutes, and you lose healthy heart tissue. So if we try to think about what's happening at a cellular level to understand this process, you can think of heart tissue as just simply a brick wall, in which the bricks are the beating heart cells called cardiomyocytes. And the mortar, which provides structure, is a protein known as collagen. So unfortunately, after a heart attack, what happens is you lose beating cardiomyocytes and you replace it with excessive amounts of scar, which is made up of collagen. This doesn't beat and it can lead to heart failure. So fortunately though, what we're studying is cardiac stem cell populations which can be tucked away within the mature cardiomyocytes of the heart. These have regenerative ability, and my thesis project is focused on harnessing the regenerative potential of these cardiac stem cells to regenerate tissue after injury. The way that I'm doing this is by using what's called the transgenic mouse model. So this is a mouse that has had foreign DNA inserted into it so that I'm able to track and identify early cardiac populations as they mature into beating cardiomyocytes. And so what I'm showing here is just one of these heart cells that I've labeled that has become a mature cell. What I see with this model is that there's initial levels of regeneration. Your heart's a pump. It has wear and tear as it beats. And so there's a regeneration of this tissue over time, but after injury, that drops off. So what I'm currently doing is I'm working on cardiac cell culture, not in the animal, but outside in a dish. And so I'm trying to optimize this pre-cardiac population in terms of treating it with different compounds to push it from this back to a mature cardiomyocyte, any pro-cardiogenic effect. What I'd like to do from these results is go back into my animal model and then treat it with this kind of cocktail of compounds that can be beneficial so that it improves after recovery. And so what I hope to do is move from what was once a repair process in which you have healthy cardiac tissue, and the blue that I'm showing here is the scar. This doesn't beat it, can lead to heart failure. I hope to push back towards the regenerative side with this kind of treatment, harnessing the natural potential of these cardiac stem cells. And the exciting thing is the translational potential of this research, because oftentimes what we find in these mouse models, we can move back into patients in the clinic. And so something that can be beneficial and pro-cardiogenic over time, we move into human trials. And ultimately, if we can regenerate or improve tissue, we can improve quality of life, as well as lifespan after a traumatic ischemic event, and then hopefully save lives with this research.